Imagine if you could regrow a lost arm or regenerate a damaged heart. While humans can barely manage a paper cut, some animals perform what seems like biological magic, rebuilding entire organs from scratch. But this isn't magic. It's cellular reprogramming, a process so extraordinary it's like your cells have a time machine. Today, we're diving into the science of biological regeneration and asking, why did evolution give some creatures this superpower while leaving us behind? The regeneration spectrum, from flatworms to salamanders. Not all animals regenerate equally. The humble planarian flatworm is essentially immortal. Cut it in half and you get two complete organisms. Starfish can regrow entire arms in months. Axolotls, the Mexican salamander, can rebuild limbs with perfect finger-like digits, even restoring complex structures like nerves and blood vessels. Meanwhile, humans can regrow our outer skin layer and repair bone fractures, but ask us to regrow a finger and our body simply gives up. We'll explore why some creatures crack the regeneration code while others, including us, hit a biological wall. Dedifferentiation, the cellular reset button. The secret begins with a process called dedifferentiation. When a salamander loses a limb, cells near the wound don't panic and scar over. Instead, they do something remarkable. They regress. Specialized cells, muscle cells, nerve cells, bone cells essentially revert to a more primitive, flexible state. Think of it like an adult remembering how to be a child. These dedifferentiated cells cluster into a blastoma, a temporary structure packed with cellular potential. This is where the magic happens. Cells forget what they were supposed to be and prepare to become whatever the body needs them to become. Positional information, the biological blueprint. But how do these flexible cells know what to build? Enter positional information, the idea that every point in an organism's body carries coordinates, like an invisible grid. Axolotl cells know whether they are located at the shoulder or the elbow, and this information tells them which structures to rebuild. Scientists discovered this by transplanting tissues between different locations. A tissue transplant from an elbow region, even after dedifferentiation, remembers it's an elbow and builds elbow structures. This positional memory is encoded in gene expression patterns and chemical gradients that act like an anatomical GPS system. Why humans lost the regeneration game. So why can't we do this? Evolution made a trade-off. Humans developed something far more complex, a massive brain, a sophisticated immune system, and the ability to live for decades. Our immune system is particularly relevant. In regenerating animals, the immune response is minimal and controlled, allowing dedifferentiation to proceed undisturbed. In humans, we mount an aggressive inflammatory response to injury. Our body essentially says, seal this wound now rather than let's take time to rebuild. Additionally, humans evolved rapid wound closure through scarring as a survival advantage in a dangerous world. Scar tissue forms fast, protecting us from infection, but it blocks regeneration. We chose speed and protection over restoration. The role of genes, what they have that we lost. Researchers have identified specific genes that govern regeneration. The xenopus frog, an ancestor of many modern amphibians, has gene networks like GATA4 and GATA5 that activate dedifferentiation cascades. Axolotls express high levels of regeneration-associated genes that humans have too, but ours are often silenced or expressed at much lower levels. We have the biological toolkit, it's just collecting dust. Even more intriguing, some humans show unusual regenerative abilities. There are documented cases of children regrowing fingertip bone and tissue. Some researchers believe our regenerative capacity isn't gone, it's dormant. The genes are still there, waiting to be reactivated. The immune system paradox, friend becomes barrier. Here's where it gets complex. Immune cells like macrophages play a nuanced role. In regenerating animals, macrophages are present, but they are in a M2 healing mode, pro-regenerative rather than pro-inflammatory. In humans, the default immune response produces pro-inflammatory macrophages that release chemicals triggering scar formation. Scientists have shown that by artificially shifting macrophages in mice to the M2 state, regenerative capacity improves. This suggests that our powerful adaptive immune system, the very thing that keeps us healthy, might actually suppress the regeneration programs that still exist in our cells. The cellular reprogramming breakthrough, Yamanaka factors and induced pluripotent stem cells. Fast forward to modern medicine. In 2006, Shinya Yamanaka discovered something revolutionary: 4 genes OCT4, SOX2, KLF4, and c-MYC could reprogram adult cells back into a pluripotent state. Essentially, he recreated the dedifferentiation process artificially in a petri dish. This led to the creation of induced pluripotent stem cells, or iPSCs. While not identical to a blastoma's natural regeneration, iPSCs prove that cellular reprogramming is possible even in mammals. Researchers are now exploring how to trigger this reprogramming in living tissue, potentially unlocking regeneration without the need for dedifferentiation's natural blueprint. Epigenetic clocks, reversing biological time. Beyond genes themselves lies the epigenome, chemical markers sitting atop DNA that control which genes turn on or off. Recent research shows that regenerating animals have the ability to reset their epigenetic clocks during the regeneration process. This is like cellular time travel. Cells temporarily reset their age to a more flexible, plastic state. Scientists led by researchers like Juan Carlos Isbisua Belmont have shown that partial epigenetic reprogramming in mice can improve regenerative capacity and extend lifespan. The process doesn't fully revert cells to youth, but it loosens the constraints that come with aging, allowing dormant regenerative programs to awaken. The scar tissue villain. Can we overcome it? A major barrier to human regeneration is scar tissue. But recent approaches are targeting this directly. Scientists have discovered that the transcription factor YAP, a master regulator, suppresses scarring when inhibited. In mice, blocking YAP allows wound healing to shift toward regeneration. Additionally, researchers are experimenting with the idea of resetting scar tissue by reactivating dedifferentiation programs within existing scars. Imagine treating old scars with a therapy that tells the scar cells to regress and rebuild properly. It sounds like science fiction, but the underlying biology suggests it might be possible within the next decade. The future, engineering human regeneration. The path forward lies in hybrid approaches. Researchers are creating biological scaffolds that mimic a blastoma's environment, structures that provide positional information while suppressing inflammatory responses. They're engineering immune-privileged environments using biomaterials. Some teams are even considering reversible immune suppression paired with stem cell therapies to encourage regeneration. The goal isn't to transform humans into salamanders overnight, but to unlock targeted regeneration, regrowing damaged heart tissue after a heart attack, restoring bone and spinal injuries, regenerating pancreatic cells in diabetics. Within 20-30 years, the first human regenerative therapies may become routine medical treatments. The philosophical angle: what regeneration means for identity. There's a curious philosophical question lurking beneath all this science. If axolotls can regrow their brains, does the regrown brain contain the same consciousness? If humans could regenerate our bodies completely, would we still be ourselves? Some regenerating animals seem to preserve memories even after brain regeneration, suggesting consciousness might be about patterns rather than matter. This raises profound questions about identity, continuity, and what makes us us. Perhaps the most fascinating aspect of studying regeneration isn't just about gaining medical powers, it's about discovering fundamental truths about how life encodes information, memory, and identity at the cellular level. The ability to regenerate organs isn't magic, it's cellular programming that some species mastered while others, like us, evolved in different directions. But the exciting truth is that our dormant regenerative genes might still be waiting to be reactivated. What started as nature's quirk might become medicine's breakthrough. If you enjoyed this deep dive into biological regeneration and the science of cellular time travel, smash the like button and subscribe so you never miss a video like this one.